So we're going to move focus slightly, and for the second part of the webinar, we're going to talk, as has been said, about whole genome sequencing for surveillance of AMR. And this reflects work that has been done over the last year and a half by WHO AMR teams, both at HQ and in regional offices, the network of AMR collaborating centres, and also a colleague from the UK called David Annanson. So you'll see some of the documents that have been produced. So we've already spoken about the functions of a reference laboratory, and Jorge spoke about phenotypic susceptibility testing. So they are the core activities of most clinical microbiology laboratories. And if they're not, then that's what GLASS aspires to get them to do, to perform high quality phenotypic testing. But of course, molecular methods can also be applied for detecting antimicrobial resistance. And this can be looking at particular genes that encode particular resistances, or increasingly could be by sequencing an entire genome of a bacterial isolate and then searching that genome sequence data for known resistance genes. So for AMR surveillance, the traditional phenotypic approach, the molecular approach, and the whole genome sequencing approach are all complementary. But if you use any one of them in isolation, you have to be aware that they have limitations. So the document that you're seeing here on the right was published on the WHO website last year, 2019, and set out to identify how molecular methods could be used to complement the existing phenotypic methods being promoted by GLASS. It emphasizes that we can detect acquired resistance genes or mutations in the genomes of bacterial pathogens by a variety of molecular methods. These molecular tests, if they're PCR, can be far faster than susceptibility testing, and they can be more sensitive in picking up markers where some results, some isolates may not express resistance at high level. So this comes on to an important caveat where we have to recognize that the presence of a resistance marker, be it a gene or a mutation, does not always reliably predict phenotypic resistance. Some isolates will have the gene, but will be phenotypically susceptible by CLSI or UCAST breakpoints. Equally, absence of a resistance marker does not reliably predict susceptibility in all instances, because we always have to be aware that there are unknown mechanisms of resistance emerging fairly frequently. So molecular tests can be used to confirm what resistance mechanisms are present in isolates that have a relevant phenotypic resistance. So you could use a molecular test for ESBLs to characterize the type of ESBL in an Enterobacteriaceae isolate that had an ESBL phenotype. And these molecular data can contribute additional levels of information for surveillance systems, and can also guide the development of new services within laboratories, within national reference laboratories. So this is one of the figures pulled from that 2019 paper, and it just shows how, for this report, the different types of molecular test at the top were then broken down by their relative cost and their relative complexity. And then they were considered for suitability to a range of different laboratories at different stages in the implementation of molecular methods. So in this example, Lab 1 is a laboratory that hasn't really used molecular methods much previously. And so there are some methods that are relatively easy to establish, and they should be focusing their efforts there perhaps. At the other extreme, Laboratory 3 in this example would have lots of experience of many different molecular methods, access to the equipment, access to the consumables needed, and so they have a broader choice in terms of what's available to them and what's affordable for them. So moving from those molecular methods to whole genome sequencing, one of the important things I would emphasize is that whole genome sequencing is just another tool. Without interpretation, it's just another method. But having said that, it's a method that will give us the complete DNA sequence for the bacteria that we are interested in from an AMR perspective. It therefore can provide a better understanding of the mechanisms of resistance in there, because you're no longer looking for particular genes. You're looking for any gene that you know exists. And also, it can be used for typing. It can now start to look at the relatedness of the isolates, the strains that are expressing resistance, and so allows outbreak investigation, national spread, international spread to be investigated and documented, as we'll see later in the talk. So these figures, again taken from the latest report, just show how numbers of publications on the use of WGS for AMR identification have begun to increase dramatically since 2013. And also in the lower graph, the skew, the bias towards the countries that are depositing these sequences into archives. So huge numbers from the US, the UK, but after that, the numbers very rapidly drop off. It would be great to see more countries submitting more isolates genome sequenced so that we have better representation of antimicrobial resistant organisms across the globe. So the purpose of this new WHO document on WGS is to present the benefits and limitations of whole genome sequencing technologies, short read and long read, for AMR surveillance. To emphasize that the analysis of that can lead to better understanding of individual bacterial pathogens for the purposes of AMR surveillance. And it also starts to set out the requirements that countries and member states will need if they are interested in building new WGS laboratories, or if they want to upgrade existing laboratories to be able to perform high quality whole genome sequencing. And again, as with the molecular paper, it addresses laboratories that are currently at different levels of complexity. So in terms of characterizing AMR for the surveillance objectives of GLASS, the phenotypic on the left will allow us to monitor AMR rates, and of course these appear in the GLASS reports, assessment of the frequency of AMR infections against the background rate of all infections caused by particular organisms. It provides data that can inform national lists of essential medicines. So is the prevalence of resistance to particular drugs compromising the essential medicines list in some countries. And of course, it can also be used to benchmark the relevance of treatment guidance and identify when treatment guidance needs to be updated because of emerging or high prevalences of resistance. So if we turn then to the right-hand side of the slide, WGS can complement all of these phenotypic methods because it gives us the opportunity to understand the genetic basis of resistance mechanisms, and also allows us to start to pick apart, to differentiate isolates that have very similar susceptibility profiles, and so are phenotypically identical, but may be different at a molecular level. An example here may be isolates that have metallocarbapenemases, some of which are NDM, some of which are VIM. It also allows us to start looking at the location of those resistance determinants. Are they mutations in the bacterial chromosome or genes in the acquired genes in the chromosome, or are they on plasmids? If they're on plasmids, are those plasmids highly transmissible? And this starts to provide valuable information on how that particular type of resistance will spread between strains, species, and genera of bacteria. And of course, as we've already mentioned, it allows you to type isolates in the same experiment. And this allows you to start identifying linkages between isolates, similarities, and provides data for the investigation of local outbreaks or international high-risk clones. So the potential uses for AMR are shown here. They can be applied at different levels, as we'll see in some of the subsequent slides. They can be used locally for detecting resistance mechanisms, for the identification of novel mechanisms. The same applies at local or subnational and national level, but here you can start to compare isolates that have been collected at different surveillance sites. You can start to understand the similarities of organisms within a country and start to look at possible transmission networks and potentially tracing sources. And if you apply whole genome sequencing to international collections of organisms, then you're now looking at population scale pathogen characterization. You can look at high-risk resistant clones. You can start to look at the impact of any interventions that are being made, and you can detect the spread of organisms across country boundaries. So for whole genome sequencing to have greatest impact on public health, it has to be combined with other data. You have to be able to interrogate the whole genome sequence to pull out the information of interest, but then you need to combine it with the epidemiological information, the clinical information, and the phenotypic microbiological data. And we've already heard today how we have to remember constantly that ultimately there are patients in beds, and even for surveillance, we are trying to generate data that will improve the treatment of those patients. Whole genome sequencing generates new insights into disease transmission. It lets us look at the dynamics of resistance and potentially lets us look at virulence of particular clones as well. And it allows this subtyping of the pathogens for characterization of spread. But as with the other molecular methods, the bottom bullet point here is critical. You cannot use whole genome sequencing data to quantify the level of susceptibility that a particular isolate will show. There is not a perfect correlation. It's not yet suitable for the majority of bacterial species, including those that are used for glass, to actually replace whole phenotypic methods with whole genome sequencing yet. The data are not sound. The correlations are not good enough. It's improving, but I don't think it's there yet. For some organisms, those predictions can be made with confidence and can actually be used to guide treatment. So here I'm thinking about TB, hepatitis C virus. But for the majority of the organisms in glass, those correlations aren't sufficiently strong. The clinical data aren't sufficiently strong for most people to take that leap to say, I would be prepared to treat my patient on the basis of WGS data alone. So this figure, again taken from the recent document from WHO, highlights how all that information, the epidemiology, the clinical data, the WGS, and the susceptibility data come together to be combined. So this links to the analytics capability that Ariana was talking about, to identify areas for public health action, where you can start to prevent infections, or if you can't prevent them, to control them. And you can start to identify community health interventions as well. So the current limitations of WGS for AMR are set out here. And also on the right, you can see the cover of this latest document, which was published in September and is available on the GLASS website. WGS is hugely expensive, even for high-income countries, if done at scale. It requires substantial initial investment in the hardware, in the laboratories. But that's not enough. You then also need to consider the workforce, because sequencing and bioinformatics are not part of the traditional skills for microbiology workforces. They're changing to be able to cover some of this, but bioinformatics is still a specialism, and we need to invest in the training of our workforce to be able to perform this on a more regular basis. We also will need the standard operating procedures. Jorge's already mentioned this in terms of phenotypic testing, but it's relevant to every method that's being used in a microbiology laboratory. If you don't have standard methods, if you don't have quality assurance protocols, then you run the risk of generating poor quality data, which will lead to poor quality surveillance. So we need to be making sure that we're raising the bar constantly and making high quality data available. And I've already said that for most pathogens, including those in glass, and for most antimicrobial agents, the predictive sensitivity and specificity of data generated through whole genome sequencing is not sufficiently high to allow quantitative measures of AMR phenotypes to be predicted with sufficient certainty. You could probably do it for some organisms on a qualitative basis, inferring susceptibility or resistance to some level, but you wouldn't be able to predict an MIC or a zone size, for example. And as we think about whole genome sequencing on a global scale, data sharing is not currently standard practice in different countries. There are open access public databases for sequences, but we need to make sure that the countries providing this data are protected and that there are data sharing agreements to make sure that any use of those data are fair and equitable. So a few examples. We'll start with local use, and as I've said, you could look at known resistance mechanisms. So you could mine a whole genome sequence for ESBL genes, for carbapenemase genes, for MEC-A, MEC-C in a Staph aureus, and for resistance factors. You can also start to identify novel mechanisms of resistance. Now, this would largely be by identifying resistant isolates from a phenotype where you haven't found a known resistance gene. And you may then ask your bioinformaticians to look more critically with non-standard methods to try and find new methods of resistance. And we can start to look at how strains are related if they come from a single center, a single hospital, a single ward, to start to identify outbreaks, and then to look at how those outbreaks evolve and subside, and to look at the impact of interventions made. But this requires whole genome sequencing in very close to real time if it's being used for real-time outbreak studies. Of course, being able to do this means that you need a functioning microbiology laboratory. You also need that laboratory to have either its own sequencing capacity or to have access to sequencing capacity through collaboration. And that must include the bioinformatics expertise. Again, that could be in-house, but it could be outsourced as well in the first stages. And it also requires reference data and tools in terms of high quality curated databases that list resistance genes, and there are many of these available. If we increase the scale of our sequencing ambitions to a national use, we can now start to look at the genomes of similar organisms in different sites. Maybe we have ESBL producers in multiple sites within a country, and we want to compare them. Again, when combined with susceptibility testing data, it can start to identify organisms that may have novel mechanisms of resistance. These we may choose then, if they're a national problem, to investigate using other sequencing methods. Long read sequencing here may allow us to identify resistance conferring plasmids, and we can start to see if those plasmids are similar in different parts of the country. It also starts to allow regional transmission networks and allows people to monitor the evolution of AMR. And there are examples here, for example, with highly resistant strains of Neisseria gonorrhea, where sequencing data has been used to be mapped onto sexual networks. It also allows sources to be identified if within your country your outbreak crosses jurisdictional boundaries, different states or different provinces. And again, it can help inform control strategies. Now, to be able to do this, ideally, this is being done coordinated by a national reference laboratory, so linking with Jorge's earlier talk. And this should have high throughput sequencing capability, ideally the computational facilities and the expertise it needs to analyze the data. But again, there is a model where some countries in the early stages of implementation may choose to collaborate with other countries to access their sequencing requirements. Again, we need highly curated databases, but if we're now looking at strains from across countries, we need to be combining it with our epi and clinical data. And we need, ideally, data processing tools and visualization tools, because if we're talking at this level, at country level, we're going to want to make very clear messages to people that won't understand the details of gene sequencing. This just shows one slide from colleagues in the Philippines, looking at how they introduced whole genome sequencing back in 2013, 2014 for monitoring the basis of resistance in a range of different organisms. This summarizes what they've been doing. So at top left, you can see that they have sites across the country that are supplying resistant isolates. They whole genome sequence them, and they then analyze them to define the types of strains. They can look at strains that are of particular interest, identify high-risk clones, and then it comes to the point that Nan and Ariana was making earlier. That data does not just sit then in the reference laboratory as research. It has to be fed back for action. So you can see here, it's fed back, the public health implications, fed back personal level to the local hospital that was submitting the isolate, also internationally, potentially to GLASS, but to the National Department or Ministry of Health as well. It has to be that flow of information. And if we look at the larger scale application of whole genome sequencing, this is international. We're now looking at related pathogens, same species, but across geographical borders and across time as well, if we have longitudinal surveillance systems. We can now monitor the emergence and spread of resistance within bacterial populations. We can detect newly emerging clones and trace how they spread within countries and between countries. We can also look at new mechanisms of resistance, and this can guide research and also can guide the development of new diagnostics and can provide the impetus for companies to develop new treatments. We can also look at the spread of mobile genetic elements that are causing that resistance, the plasmids, the transposons, and how they work and move to new locations. And we're looking now at this on a global scale. So clearly this needs multiple laboratories, ideally networked and sharing information regularly. They need trained personnel, and we need large-scale structured surveys to make the data that are being generated meaningful and comparable. This needs extensive databases and, as I've already mentioned, needs that equitable data sharing agreements, which comes on to Nagoya and so on. So this is one example from the EUScape project that David Annenson ran, looking at carbapenem-resistant Enterobacterales across multiple European countries. And if we just look at the map on the bottom right, it starts to show how the visualization helps explain what's going on. So the green dots, the KPC producers, are far more geographically clustered than, for example, the pink dots, which are the OXA48 producers. So you can see that the OXA48 producers are far more widely scattered. If you look at the dendrogram on the right of the slide, then this shows how you can start to analyze the KPC producing Klebsiella pneumoniae, and you can start to break those down into the sequence types, the clonal groups, and start to identify the high-risk clones and understand the biological basis for the resistance problem that's being faced in many countries. The document also goes into the requirements for whole genome sequencing. So it talks about the timing of the introduction. Now, clearly, every country will consider introducing whole genome sequencing into its armory of AMR techniques at different times. They have to do it when their country is ready or when they are prepared to collaborate with other countries to provide the capability. They need to consider the infrastructure requirements. They need the laboratories. They need the computational capacity. They need the bioinformatics skills. Do they go down open platforms, free to use, or do they go for proprietary solutions? Even the bioinformatics options, there are some companies offering cloud-based solutions where sequencing is uploaded and analyzed and results returned. We also need to be making sure of the quality. Jorge's mentioned that for the phenotypic. It's exactly the same for the genotypic and the sequencing as well. Rubbish in always means rubbish out. So we have to make sure that the data are quality assured, controlled, and that they conform wherever possible to international standardization, and that we are using methods that are benchmarked against international standards, which don't yet currently exist, but are in development. And then we also have to consider the procurement problems. And this is just one slide that summarizes the problems of buying Illumina MiSeq instruments in a number of countries compared to the UK. So you can see at the bottom of the slide, it would cost about £140,000, US dollars, to buy this machine. In some of the other countries on the table, it's 2, sometimes 5 times as much to buy exactly the same instrument. And then they do not necessarily have the same supply chain for regular supply of reagents. And it may be more expensive even to buy those reagents. So this is, again, something that needs to be addressed as we start to see whole genome sequencing used more widely in different countries. We also need to consider the training of the staff, which I've mentioned, and the data collection, how it's going to be shared, how it's going to be stored, how is it captured, how does it feed into GLASS, how can we use WhoNet, other softwares to get those data into the GLASS platform. So in conclusion, whole genome sequencing gives us the opportunity to enhance how laboratories investigate AMR for surveillance and for research at the current time. It's not yet there for the GLASS pathogens for treatment at individual patient level. It gives us the advantage of being able to detect all known resistance genes, indeed all known genes. And this will allow us to understand the spread of AMR. The limitation is only in the time that we have available for the analysis of the isolates. But as with any new technology, we have to consider what the resources needed are against and balance these against country needs. And we need to be considering when the most appropriate time for launch in different countries actually is. Thank you very much for your attention.